There are 6 million of these cytotoxic T cells in just 1 tablespoon of your blood. Their job is to crawl through your tissue and feel the outer surface of cells to try to identify those that are infected with an intracellular pathogen, such as from a virus, a bacteria, or a parasite. Once they find one of these cells, they slow down and they wrap their membrane around the outer surface of the compromised cell. And they're trying to detect the presence of proteins that are foreign below the cell surface. Because the presence of foreign proteins inside of this cell reveals that this cell is making a pathogen, and this earmarks it for destruction. Let's learn what molecules are involved in this process. Below the cell surface, proteins are constantly being produced. Now, the vast majority of these proteins are self-proteins. So these are proteins that should be inside the cell that are completely normal. But if a virus comes along and infects the cell, it will start to manufacture viral proteins in addition to self-proteins. Peptides, which are little samples of proteins that are loaded onto a molecule called human leukocyte antigen. And they are presented to T cells that crawl through the tissue. And the T cell receptor of these cytotoxic T cells physically interacts with the HLA molecule that in combination with the peptide. And if this peptide here is foreign, it will send a signal into the T cell that will result in the release of cytotoxic granules that will destroy the cell and obviously any intracellular parasite or bacteria or virus that's inside. So let's take a closer look below the cell surface and learn how these peptides are loaded onto HLA. So on the right, I'm showing a viral protein. And this protein has been damaged, misfolded, and it has been marked for destruction by a multi-protein complex inside the cell called the proteasome. So you can think of the proteasome as the garbage disposal of the cell. It takes the protein, chops it up into small fragments called peptides. Now these peptides can make their way into the lumen of the endoplasmic reticulum, where they're loaded onto an HLA molecule. And this HLA buds out from the endoplasmic reticulum into a vesicle, which fuses with the plasma membrane on the outer surface of the cell. So cytotoxic T cells crawling through the tissue use their T cell receptor to physically interact with the HLA and peptide in combination. Now, if there is a strong binding between the TCR and the HLA molecule, this will cause a signal that will release cytotoxic granules into the infected cell, resulting in its destruction. Epstein-Barr virus is transmitted between humans through the saliva. From here, the virus will infect epithelial cells. It will replicate, where it will go on to infect neighboring uninfected cells. And it can also infect B cells, which is a white blood cell that can produce antibodies. And it's estimated that 90% of the world's adult population is currently infected with EBV. But the majority of these individuals that are infected develop an asymptomatic infection, so they don't even realize that they're currently infected with EBV. And then a subset of this 90% will develop a symptomatic infection called infectious mononucleosis. And once you're infected with the Epstein-Barr virus, you are infected for life. Now let's take a closer look at the Epstein-Barr virus life cycle, because it's a little bit different from other viruses such as SARS-CoV-2. Epstein-Barr virus infects a host cell once it attaches to a receptor on the surface of the cell. This leads to binding and fusion of the virus with the plasma membrane. This viral capsid here is degraded, resulting in the release of the genetic material of the virus into the cell, where it translocates into the nucleus. Now here is where Epstein-Barr virus is a little different than most viruses. This circular ring of DNA can reside in the nucleus for the life of the cell. It can sit quietly and not make any more copies of itself. And this is called latency, or viral latency. Now, if it doesn't progress from here, this is considered a latent infection. But under moments of stress, such as from infections, genes can be turned on, creating EBV mRNA strands, and the EBV mRNA is exported out of the nucleus where the translational machinery resides, so it can convert the mRNA code into Epstein-Barr virus proteins that are assembled into viral particles where they are released from the host cell to repeat the life cycle. So the ability of cytotoxic T cells to use their T cell receptor to detect cells that have an EBV reactivation event occurring below the cell surface is incredibly important, because if this can be detected early on, there can be signals released from the cytotoxic T cell to the infected cell to result in its destruction and preventing EBV from escaping and infecting other cells. However, the inefficiency of this detection mechanism, usually through genetic predisposition, can increase the likelihood of developing a range of different chronic conditions, including multiple sclerosis. Now, this leads us to the results of a recently published study by an Italian group where they discovered that having HLA-E variant called 0101 plus a documented case of infectious mononucleosis increased the risk of later developing multiple sclerosis by a significant amount. So, for example, those who had just one copy of this HLA-E 0101 variant had a 74% increased risk of later developing MS compared to individuals without a documented case of infectious mononucleosis. So just having the HLA gene alone did not increase the risk of MS. It was only having the gene plus having a documented case of mono. And individuals with two copies, all right, so two copies of the gene, the HLA gene, had a 201% increased risk of later developing MS compared to those without a documented case of mono. So what might be explaining these results here? So there's a couple of different explanations. So the first is that having this HLA-E variant reduces the cytotoxic T cell's ability to effectively detect the EBV reactivation event inside the cell. And that reduces the cell's ability to control latent viral infections, or reactivation, I should say, putting a lot of pressure and stress on your immune system, leading to the development of autoimmunity. Could be that the mechanism is dependent on something called molecular mimicry. So this happens when a self protein is confused by the immune system for a non-self protein. So in the case of MS, myelin is a self protein, and it can be confused for EBNA1, which is an Epstein-Barr virus protein. And so this can lead to molecular mimicry, in which the T cell that is trained through its development of its T cell receptor to recognize a very specific combination of HLA, in this case HLA-E 0101, in combination with a very specific peptide or protein sample from EBV. If it thinks that this myelin, which will be presented in the central nervous system, is actually an EBV in a antigen, then it might mistakenly think that that myelin sheath producing cell is actually infected with Epstein-Barr virus, causing it to become attacked, destroying the myelin sheath surrounding the nerve, causing the pathology of MS. So that could potentially be what's at play. Of course, this is speculation, and we do not know the exact mechanism. It may have nothing to do with EBNA1 or myelin at all. It could have to do with something completely different, but that is just a mechanism to potentially explain why this might be specific to MS and not other autoimmune diseases. So we had a couple questions on social media that were very similar. So I'm not going to feature every single question, but they were all related to the question that was posted here. And this was from an individual explaining that they had EBV from childhood with a documented case of mono. And they later developed POTS, long COVID, and had a potential MS scare. Then the question that they have was, can my doctor test for this gene? And then what needs to be done? So testing for this gene to determine whether or not you have this variant is not something a physician would do because it wouldn't necessarily change the course of treatment. So it's not like there's technology that can swap out certain variants that are associated with disease. So it's not as if there's technology that would allow you to swap out this 0101 variant for an 0103 variant, which has not been associated with development of MS if you have mono. Um, but what it does do is it really teaches us about the biology of Epstein-Barr virus and what could potentially be going wrong to lead to the development of specific autoimmune diseases such as MS. So it teaches us more about the biology. But what I would say is if you have an array of chronic conditions and you have a documented case of mono, or you may think that you have EBV reactivation issues where you have this robust chronic inflammation, there are tests that your physician can perform to determine whether or not you have EBV reactivation in your blood. So there's markers they can look for to determine whether or not you have chronic reactivation issues. And there's other drugs out there that are not necessarily licensed to treat EBV. There's actually no drug that's licensed to block any stage of the EBV life cycle, including a vaccine. But there are drugs that can be used to limit its reactivation and try to control the replication of the virus so that you don't put a ton of chronic stress on your immune system to uh, uh, confuse your immune system and create the development of autoimmune conditions or other post-infectious chronic conditions. So if you haven't already signed up for our deep dive series, this is a wealth of information specifically about EBV and the virus and different aspects of it. But one of the sections in here talks about therapeutically targeting Epstein-Barr virus. There's a table of different supplements that you can access yourself, and there's also drugs that you could have a physician prescribe if there is any evidence that you have issues with controlling your Epstein-Barr virus infection so that it doesn't progress towards full-blown MS or some of these other chronic autoimmune conditions later down the line. So with that, we thank you for listening. If you liked the video, please give it a thumbs up, like and subscribe, and please share this with others. And thank you for listening.